Your reading assignment to go along with today's lectures was about the importance of Drosophila in helping us to understand genetics, cellular biology, chromosomes, genes, all of the amazing discoveries that have been made in flies. And flies have been kind of the workhorse, if you will, of model organisms. And they have been incredibly useful. But like any model organism, there are limitations. So depending on what you want to do, you may have to pick a different model. So for instance, although fruit flies serve as a good model for eukaryotic cells, they may not be the best model if we're studying something as detailed as mammalian immune response, because that's evolved to be quite different from what happens in the arthropods. And the same thing is true at the developmental level. But again, remember, because of remarkable amounts of conservation at the developmental level, there's a lot more that we could do in Drosophila than we might otherwise have thought at the outset. Because, for instance, we could do eye development, at least early eye development, and look at some of the things there. Now, I'm going to make a case that despite the really, really important role that model organisms play, for whatever study we're doing, if we want to understand it in an evolutionary context, we need to expand beyond model organisms. So let's address why Drosophila was chosen, and then we'll talk about the advantage of looking beyond a model organism approach. So some of it is just chance because, there are probably many other fruit flies. Melanogaster is the main one that was used, Drosophila melanogaster, but there are other species of fruit flies that are just as easy to take care of and work just as well. So there's a little bit of just chance, whatever ones they were most common in the area where the people started working with them, and they were easy to keep in the lab. But like most model organisms, Drosophila are small, reproduce rapidly, and are very easy to keep. They're not, don't require a lot of care. They will last for months and months if you pretty much just provide a little bit of food and then recycle their colonies every once in a while. So to some extent, it's happenstance, but they also have many characteristics that make them ideally suited for a model. But there are other insects that could have fit as well. Now, one of the weird things about Drosophila is it's in a kind of a specialized developmental niche. It lays eggs in fruit, which is rotting, and it's a very ephemeral food source, meaning it doesn't last very long. And so it's got some very dramatic changes. So remember we talked about heterochrony, changes in timing. It's got kind of a dramatic compression of its developmental series. It develops much quicker than most insects because if it didn't, the embryos would die before they could take advantage of their food source that is rotting and going away. And so that makes them perhaps not ideal as an evo-devo model. That doesn't mean they're still not useful. There are many things they still do the same, but we might expect them to have evolved some changes in development because their period of development is so compressed and different selection pressures on it compared to most other arthropods. Okay, so just that, but that applies to all models. We need to recognize their limitations where studies in that system might be broadly applicable and where they might be more narrowly applicable maybe to a small group of organisms. So we really need to expand beyond Drosophila, and people are doing this and have been doing it for decades, and so we're getting dramatic work. This has been especially facilitated by our ability to sequence entire genomes and transcriptomes and do work on a scale at the genetic level in organisms that are not easy to work with. So mammals as a general group are very difficult to work with for evo-devo because embryos are held internally. You have to sacrifice the mother and the embryos to look at them. It's really tricky. But lots of work is being done and it's being expanded beyond that. And again, there are also many ethical considerations. There are other organisms amongst the arthropods, even amongst the insects, and we'll be talking about those, that really are incredibly difficult to work with as in embryos also for different reasons, because they can't be kept in captivity, or they lay so few eggs, or they have a weird life cycle or limited distributions. There are many, many other reasons. So we can understand a lot generally by looking at model organisms, but we need to do some of the hard work and expand beyond the model organism system if we want to understand big picture. So here's a summary of the level of animal diversity that I expect you to know for the upcoming exam, right? So choanoflagellates, sponges, cnidaria. We then have this big group called the Bilateria, and you should know the two major groups, the deuterostomes, protostomes. And then within each of these groups, this is maybe add this in your notes, right? In the deuterostomes, we have the vertebrates and the echinoderms. Those are the two major groups, and there are other subgroups in there, but or maybe even better, chordates, which is a little bit more inclusive, includes the vertebrates and a few other closely related things. But we have the chordates and the echinoderms and a few other minor groups. Here we have the ecdysozoa, which are represented here by these major groups, and then the Lophotrochozoa. So know that level. Good review there for your phylogeny and the level of animal relationships that you need to know. Okay, but there's a massive amount of diversity. Primarily, most of this is in the Bilateria, but there are important differences that if we just focused on the Bilateria, we would miss. So we want to include these others, cnidaria, sponges. Of course, we could also throw in the ctenophores, which are a little bit more controversial as to how they are related to these other ones. And then we talked about the Placozoa also, which are more recently included. But anyway, this level is a good outline for what you need to know. So arthropods are a very, very good group to study diversity because there's just so many amazing forms and tons of diversity, even within small groups. So we have the crustaceans represented here by this mantis shrimp with lots of cool phenotypic differences from other crustaceans. We have the hexapods, including many winged versions, and we'll be talking about those in a lot of detail. This is the closest living relative of the arthropods, which is shows some similarities, but some important differences, and so we could begin to study those and look at how those differences came about. And of course, humans, when I first put this together, this dates when this lecture was first originated, right? I could pick any human. Maybe I should pick like Albert Einstein or Gregor Mendel or somebody who's a little bit more, I guess, related to the scientific field. But, you know, any vertebrate we could put in there, right? And so we can study them, and it's only when we are able to look at these in context. Now, obviously, working with humans, there are massive, both logistical and ethical barriers to looking at human development, and that's really, really tricky. But if we can look at perhaps mouse development and rat development, maybe expand beyond rodents and understand that and see similarities in distantly related groups, we can make extrapolations and understand that probably we're going to see the same thing in humans. So it's worthwhile looking at an evolutionary perspective because it takes us from a single model organism with maybe lots of question marks as to whether what we're seeing in that model applies to everything, or maybe it's something that evolved and is specific to just a small range of organisms. So an evolutionary perspective is very, very critical and allows us to expand beyond what we can do in models, but models are very important. Okay, so fruit flies, relatively easy to take care of. We're not going to go into the details. We do a little bit of this when I have a wet lab. We use Drosophila as the primary organism, but you need to understand how we began to identify genes in Drosophila. This was before we could look at them on chromosomes, although that came fairly quick in this new synthesis of biology as we begin to put Mendel's ideas and Darwin's ideas together and understand what's going on. Okay, but initially, they would raise tens of thousands of these in colonies, and then they would usually use x-rays. Sometimes there were chemicals, but they would usually use x-rays to have some elevated level of mutation. And then they would screen these tens of thousands of babies that could be made relatively easily and cheaply in the lab for mutations. And they would find these mutations, that had maybe smaller eyes. So this is what a wild type fruit fly looks like, or changes in shape, or loss of eye altogether. And so they could identify different mutants, and then they could breed them together to determine whether it was a dominant mutation or a recessive one, if it was sex-linked. That's all genetics and kind of background, but not really critical for this class. And so they were looking at these, and some of these became identified as these key mutations, these homeotic mutations, right, where we would have complete changes into the body plan of the organism. And so that became identified very early on as a key target for study. And so this is a UBX mutant. UBX is a really important gene. We'll use it over and over again in this class as an example. And then there were others that were not as fundamental to body plan, but still played a role. So here's one that plays a role in wing development, where we get this little funny formation of the wings, and it has maybe some other ramifications on body form and type also. And so we begin to identify early developmental genes, genes that are part of the toolkit, but maybe a little bit later on, right? So eyeless gene completely eliminates the eye, but a later gene in eye development might just simply change the shape of the eye, right? And so we can begin to identify these and maybe even put them to a little bit in the context of when they occur in the developmental series of the organ or the structure that we are looking at, okay? So let's look at what we begin to understand about the genetic toolkit of Drosophila. And we're going to start with these homeotic mutations. Now, it's important to realize that these homeotic mutations occur in all organisms. So there are examples, even going back hundreds of years, of descriptions of these in a moth, for instance, where you have an antenna that looks a little bit like the end of the leg. Invertebrates, in mice, where we have there's usually a very specific number of thoracic vertebra, but there's some rare homeotic mutants that have extra thoracic vertebra. And these were, again, they occur at some level in nature and are found periodically, but the key advantage of the model system is that we can dramatically ramp up mutation. That means lots of organisms are going to die, but that's what the trade-off for this. We can dramatically ramp up the mutation rate and identify a much, much higher level and identify interesting homeotic mutations that maybe would be 1 in a billion in nature. We can maybe ramp them up to 1 in a million or 1 in several 100,000. And so if we screen enough fruit flies, we're going to start identifying and finding these. And then once you've found one, if it's a survivable mutation, you can make these lineages, these strains of fruit flies that have those mutations. And if we release them into the wild, they'd almost certainly die and not pass on those mutations. But in the lab, we can keep them separate, give them a little bit of an advantage, treat them nicely and carefully, and we can get some of these survivable homeotic mutations to create distinct lineages in the lab, okay? Now, many of these could be identified to a specific type of mutation. A loss of function, meaning a gene that was not expressed in the correct area. We just turn off gene expression. Or ectopic expression, so we're getting genes that are being expressed in the wrong location, right? And so, although initially we knew them as homeotic mutations, sometimes we didn't know the exact mechanism of that mutation. Now, the first question that people were looking at was, how can a change in one gene result in such a dramatic impact downstream? And it didn't take people very long to realize that it really depends on the sequence of development and where the gene that we have mutated is involved. So in some ways we can make an analogy of building an animal a little bit like building a house. If you were to change a process very early on in building of a house, you might have a very dramatic impact in the overall structure and function of that house. Whereas if you did it at the very, very end, like, we're going to change the paint color, it's maybe a fairly minor change. And the same thing applies to developing animals. If we mutate something that is involved very, very late, like adding bristles to the end of a wing, and we express it in the wrong place, we get bristles on, the other structures. This, by the way, is a good thing to know. Fruit flies and other true flies come from the order Diptera, which translates as 2 wings. Most flying insects have 4 wings, which the mutant version kind of reverts back to that more ancestral form. But wild-type fruit flies and other true flies have their hind wings modified into these structures called halteres. They work like little gyroscopes, and so they're primarily sensory, and they don't provide lift during flight. So if we mutate a gene very early in development, we end up with complete wings, because not only did we change that one gene, but we changed all the downstream targets for that gene. However, if we change something much later in development, like the development of bristles on the end of the wing, then we might see only very minor changes into that structure. So genes that are early in development are going to have a much more severe phenotypic impact than genes that occur later in development. And this also led us to this idea of cell autonomy, and this reinforced the ideas that were established by those Mangold and Speeman experiments, right? Where if we make changes early in development, we can dramatically change the fate of not only the cells that we're changing, but everything downstream. They can act as signalers, right, to send out morphogens and change the fate of neighboring cells as well. And so we can look at a very early embryo as basically a blank slate with waiting for signals to occur to tell the cells what to be. And we're going to look at a couple of the very important cellular pathways that give those signals in Drosophila in the coming lectures. So, UBX was one of the earliest. UBX and Antennapedia were the first two, but later we discovered this core set of homeotic genes that we're going to just call Hox genes for short. The Hox genes are a very specific subclass of developmental genes that are critical for patterning the anterior to posterior development of all animals, but they were first discovered in the fruit fly, and then later we found homologs, orthologs, and paralogs in all animals. So let's look at the homeotic genes. You should, because the tests are online, you don't need to memorize these, but you should be aware and have easy access to this, right? So there are 8 canonical. Now, you'll see why I say that in later lectures, but basically canonical means part of the core key set that are all serving the same type of function. So there are 8 canonical Hox genes in Drosophila, and they are found on a single chromosome in the same order that they are expressed in the developing fruit fly. Okay, so labial is expressed in the most anterior part. Here we've got them in the developing mouth parts. Proboscipedia and deformed are then the next most anterior, and we're going to work our way all the way back to the posterior part of the animal. Okay, so here are the names of these 8 Hox genes. We've already mentioned Antennapedia and ultrabithorax. And notice that Antennapedia, which is here in purple, is important for developing legs in the thorax. This is a bit of a simplification. So they're actually involved a little bit in the front leg and the hind leg, but especially in the middle section. And that UBX is important for this hind segment of the thorax, and it therefore is responsible for a development of the haltere. So if we mutate UBX so it's no longer expressed, this segment, which normally develops a haltere on it, develops a fully formed wing. So that would be like a loss of ultrabithorax function. So now we get this idea that ultrabithorax is serving to somehow modify a wing into a haltere. Okay? Now, a couple of things to recognize here. The location in the genome corresponds to the body segment for which these genes are in charge of development. And that came as a huge surprise. So there's some sort of a kind of group regulation that is going on to maintain order of these key genes in the genome. Okay, this is called collinearity, and was unusual because typically there's a lot of flexibility into where genes are located. You can move a gene from chromosome 1 to chromosome 2, and as long as you move all of the control mechanisms with it, so it can still be turned on and turned off as needed, often there's very little impact to moving it from one area to another. But the conservation of the order of these Hox genes across large swathes, in fact, pretty much all animals with some minor modifications, the conservation of the order tells us that there's some really important regulatory function of having them in this same order. So that's collinearity. You should know that as a definition, and it was found in Drosophila and then extended to other non-model organisms. Now, key to us understanding and beginning to explain these genes was our ability to look at gene expression. Look at either mRNAs or proteins, and you can do either. If we look at an mRNA, it's called an in situ hybridization. If we look at a protein, it's called an antibody stain. But both of them are methods where we can use, in this case, fluorescent markers to look at expression of genes. So here is a map of 7 of the 8. I think the only one that's not here is Proboscipedia. But they were labeled 7 of the 8 Hox genes in Drosophila and where they are located. And you'll notice that, as I said from the diagram here, this is a little bit simplistic. So notice, for instance, UBX is found in the hind segment of the thorax right here. But it's also found a little bit moving back towards the rest of the abdomen. Okay, abdominal or Antennapedia is found in the middle, but it's also found a little bit in the third segment of the thorax. Okay, so there is overlap of these genes, and this has become an important concept later on. So if you think about it, if we have 8 different genes, you could look at that as 8 different signals, all signaling a different specific cell fate. So we might have 8 specific body regions that could be signaled by those 8 regions. However, if we have overlap of genes, then suddenly we have dramatically increased the number of potential body regions for which we have different signals, right? So antennapedia might be one fate for, this is going to become the middle. If we have a group of cells that is expressing both antennapedia and UBX at some levels, then we might have a different fate, and then UBX could be a third. So with just 2 genes, we now have 3 different potential body regions that are signaled because there's overlap. And so these genes specify identity, and the Hox genes specifically pattern different body regions looking at the anterior to the posterior. And so that's something that was discovered in fruit flies and basically applies to all bilateria. It's a little trickier once we get outside of the bilateria, because then we have, different body axes, right? So the body axis of a sponge is very different than the body axis of a bilaterian. But in the bilaterian, we have the same idea of collinearity and all of them expressing. And if we change areas, so if I move antennapedia and ectopically express it in the head, there's our mutant. So this mutant represented a mutation that ectopically expressed antennapedia in the wrong area, right? In UBX, the mutant was a loss of function where we turned off UBX expression, and then we end up developing a wild type, I'll go back here, a wild type phenotype in not only the first thoracic segment, but also in the second one, which normally looks like, wild type just having these halteres. Okay? And we begin then to, and this is going back in the late 20th century, we begin to identify the genetic sequence that formed these. And what we found is that in one fairly short region of the Hox genes, there is remarkable conservation. And I showed you an alignment like this in a previous lecture, but here we have all 8 genes of the Hox complex. Just to be clear, a complex, we call it a complex because they're found all linked together in the same order in the chromosome. So a Hox complex refers to that order of the 8 Hox genes in the Drosophila genome. And when we take each of these and line them up, they're very, very clearly homologous areas, right? Now, if we go downstream and upstream of these genes, there's a lot of differences. So this is a really cool example of both conservation of function and neofunctionalization due to the changes that have occurred upstream and downstream of this very conserved area. Okay. Now, as you might expect, this conserved area is critical. It's at the core of the functionality of these Hox genes. So Hox genes function as transcription factors, which means the final protein product of this gene needs to bind to a target sequence of DNA so that it can impact expression of those target genes, right? So this is binding to those cis-regulatory elements that are close to target genes. And so this incredibly conserved area is the DNA binding motif. Now, this is actually present in many, many genes. The Hox genes are a part of a much, much larger superfamily of homeotic genes. But we're going to use Hox as this kind of core 8 genes in Drosophila. We'll look at them in the vertebrate in coming lectures. Okay? And so it's present in many, many other gene families that are not part of the canonical Hox genes, but are all part of the larger gene family. So here we have this idea of paralogous copies, duplicated copies of genes that are all part of this larger gene family. Okay? And so as we expand the gene family, other members of it include islets, right, which we've already talked about. And so I'm going to introduce one other phenotype, right? Mutant phenotypes of islets, depending on how severe the mutation is, are missing or mostly missing eyes. If we ectopically express islets genes, so here's an example of an ectopic expression of this key developmental gene in the antenna. We end up growing eyes on the antenna. So loss of function mutations, ectopic expression mutations can both help us to determine the function of genes. There are 3 other genes that I'm just going to introduce now. You can tell a little bit by their names. Some of them are better than others, but let's introduce them now. Distalis is developed in, or is expressed in developing appendages. And in all appendages, this is a remarkably conserved function across all animals, where anytime you're growing an appendage, whether it's a leg in or an arm in vertebrates, whether it's a segmented antenna or a segmented leg in arthropods, or even tentacles, even extending all the way to tube feet in echinoderms, these little tiny feet on the bottom of starfish, they all use distillus as the initial gene to say, hey, grow an appendage here. So another great example of deep homology. So here it is in a developing arthropod in blue, developing in early appendages. So distillus is appendage, and the name comes from the mutant version, which of course you don't grow the distal elements of the arm. And we saw something similar to that in the example with the chicken wing when we were looking at the apical ectodermal ridge. Vestigial is one that's involved in early wing development. So mutants in vestigial grow these little tiny nubs rather than fully formed wings. So that's where the name comes from. Engrailed is a segmentation gene. So we see it expressed in early developing segments. The name's a little less intuitive for that one. Just know segmentation, make a note of it. And then the last one, and the reason I'm mentioning these genes, we're going to talk about many of them at later dates, but they're all good examples of what we talked about here, this big superfamily. These themselves, eyeless, distillus, vestigial, engrailed, and apterous, are not themselves Hox genes, but they're part of the larger superfamily of these homeodomain containing genes that all serve an important developmental function. Engrailed does segmentation. Apterous, as the name might, apterous means without wings, but the name implies this important early wing development form, and so mutants in this in the fruit fly didn't develop wings. So just review, I guess there are 5 of them, right? Other examples of homeotic genes that are not part of the canonical Hox genes themselves, and they play a role a little bit later in development, but still have an important morphological impact if we were to change their expression, like express them in a different area, or if we turned off their expression.